Thank you, Bob, and thanks to the Gulfers Against Cancer. It's an incredible organization. I'm just learning about it, but 19 years of supporting cancer research. And one of my colleagues from Baylor said how important this money is to get new projects started that otherwise wouldn't be funded by National Institutes of Health. And thank you for helping us take the first step in a bold adventure toward a whole new field of therapeutics. I won't be talking today about a pathway or a receptor or a particular approach. I'm talking about a whole new field of therapeutics, which we call RNA therapeutics. And let me explain that to you just briefly. You all know that DNA is the blueprint of our bodies. Everyone's DNA is different. Your DNA is in every cell of the body. The blueprint is very important. It's protected by many mechanisms in the cell. So DNA blueprint, you want to keep that pristine. Your body, to make proteins, makes a small copy of that DNA. It's called RNA. That copy only lasts for a short period of time. It lasts long enough so that you can make the protein that the cell needs to function. Get us over some obstacles.
And we're very thankful again for your contributions, for your efforts, for everything you do. I mean, you're volunteers. You're taking time to make a difference in science, and we appreciate that. I am an alumni of MD Anderson, and I also worked in immune cell therapies, developing the processes that allows these cells to be used in a clinic. But I am a molecular biologist, so we're combining that knowledge, and we have an amazing team and amazing support from Methodist Hospital that allows us to, on the 10th floor of that building, create molecules, understand these molecules using preclinical studies, and then we take that knowledge and we move to the 12th floor, and we have GMP facilities that allow us to make clinical grade molecules for phase 1 studies.
On behalf of Golfers Against Cancer, I'm pleased to present this grant to the Houston Methodist Hospital.
Thank you very much.
Thank you very much.
Our project this year actually focuses on a rather unique observation that we made using access to some of the genetic technologies that are available at Baylor. And this observation is the fact that some of the sequences and transcripts in ovarian cancer that most powerfully predict outcome are actually not human, they're viral. They're not random viral sequences. In fact, they're very unique subsets of viral microRNAs that share a very common and specific function despite the fact that they come from 3 different viruses. My lab has already received some grant funding to explore the mechanisms by which these viruses potentially contribute to ovarian cancer and perhaps make it more aggressive, and how we might be able to exploit that insight to develop new therapies. But the purpose of our proposal to the Golfers Against Cancer is actually to explore a little bit different take on this, because our observation also opens the door to the fact that viral infections of the fallopian tube may contribute to the origins of this disease. And this is essentially an unexplored area of human biology. The microbiome of the fallopian tube is uncharacterized, even though it essentially is connected to the outside world.
And I've been associated with Golfers Against Cancer for the past 3 years now, and it was Sally who first met with us, and so we're going to miss her greatly, and grateful for all that she did and all that you have done to help our research forward. So maybe a quick update on our work. So in the past, my lab has worked on breast cancer, and but when we moved here to Houston, I thought, well, let's do something a little different than what we've been doing before. And so we turned our attention to pancreatic cancer. And for those of you that follow the statistics coming from the American Cancer Society, you probably noticed that for this coming year, the estimated number of cancer deaths due to pancreatic cancer is actually going to slightly surpass breast cancer, even though the number of cases is only a fraction of the 200,000 or so cases of breast cancer. So about 40,000 cases for this year. And for me, the most striking statistic might be the 5-year survival rate when you compare pancreatic cancer to the other major cancers. And so my group has been working on a class of genes and molecules in the cells called the nuclear receptors. And we focus on one of those. It's called the liver X receptor. And it's a receptor that's been shown to function in metabolism, both at the cellular level as well as at the organismal level. And in fact, this receptor was originally identified and developed as a target for treating heart disease and diabetes. But for those of you who do cancer research, I think you also appreciate the fact that metabolism is also a very important part for cancer cells. And so we decided to look at the function of liver X receptor in pancreatic cancer. In fact, our first project, which has been published already, was funded by Golfers Against Cancer. And we were able to show that by targeting liver X receptor, that we can slow down the growth of not only the cancer cells, but also tumor formation in...
Basically, the collaboration study is that Dr. Frago is working on prostate cancer. He's working on the cancer metabolism. And for some of you may know that actually cancer have some unique metabolism. They have a sweet tooth. They like to drink a lot of sugar, glucose. They consume a lot of glucose. And then so my lab is working on the cancer immunotherapy. So especially we are modifying T cells. We use a technology called CAR T cells, which is a chimeric antigen receptor. So what we did is that we inserted a gene in the T cells to force them to express a molecule so that they can start to search for tumor cells and to attack the tumor cells. So that one has been recently generated some really exciting clinical results. So apparently those T cells, they need to drink a lot of sugars as well. So once both of us realize what's happening, so that has come to a scenario is that tumor cells, they're consuming a lot of sugars. And then T cells, in order...
I'd like to present the Golfers Against Cancer grant to the University of Houston.
I'd like to present the Golfers Against Cancer grant to the Baylor College of Medicine.
Great. Thank you very much.
Triple negative breast cancer is a very aggressive form of cancer. There has limited treatment option for this group of the cancer. So in our study, we will use modified patient's own immune cell to fighting with cancer. So previously, we discovered a high specific tumor antigen called NY-ESO, which is expressed 50% of all kinds of the cancer. Particularly, they express 30% of triple negative breast cancer. So we will use preclinical model of triple negative breast cancer to demonstrate our modified T cell can recognize and destroy the triple negative breast cancer which express NY-ESO1.
Research-wise, this has been a great year. As you know, with your support, we've been conducting a small trial of some 5,000 women to see whether our strategy of watching CA-125 each year, and if it goes up, getting an ultrasound and acting on that ultrasound if it's abnormal, would be specific enough. We've now had 18 patients go to operation. 12 of them have had ovarian cancer, and 9 of the 12 have been in early stage 1 or 2, where it's 70 to 90% curable. As important, there's been a trial in parallel in the United Kingdom using exactly the same strategy, with 50,000 women, 10 times as many women, and 100,000 controls. This last December, they reported out the results of that trial. And it turns out that you have to wait up to 7 years for the effect to kick in. But once it does, they're on track to have a 20% reduction in mortality. That means we could save 1 out of 5 women with ovarian cancer. Now, there are still broad confidence limits, talking technically, around that 20%. And it's going to take another 2 or 3 years to see if it's really 20% or if it's 15%, and whether it's going to be practical to be able to screen. But I think the important thing is it means we're on the right track.
I am so very happy to present this check for the grant from Golfers Against Cancer to MD Anderson for their cancer research.
Thank you so very much.
Thank you. Thank you very much.
I've been working in prostate cancer for 23 years, and it remains a stubborn problem, just like many other cancers. Even though we have made some progress in the mortality, there's been about a 10% decrease in prostate cancer mortality. Nevertheless, it's still the second leading cause of cancer death in U.S. men. And from a global perspective, it really is a major killer of men. And what my research is focused on is a somewhat novel area in that what we're looking at is the interaction between the cancer cells and the nerves in prostate cancer. And I think it's generally appreciated now that the environment around the cancer cells obviously have many mutations and alterations that contribute to their malignancy, but they only grow within an environment, the tumor microenvironment, that supports their growth. And one of the things that's become clear is that in prostate and in some other kinds of cancers, like pancreatic cancer and melanoma, that there is a interaction between the nerves and cancer cells that can promote the malignant behavior of the cancer cells and provide therapeutic resistance to the cancer cells. And so we're focused on a particular signal transduction pathway that has already been shown to be very important in thyroid cancer, and there's actually an agent that's out there in the clinic for treating thyroid cancer, and it's very effective in thyroid cancer, targeting the signal transduction pathway. So what we want to do moving forward is to use this agent to see if we can improve outcomes in men with prostate cancer, both as in men with advanced prostate cancer as well as men undergoing radiation therapy. Because
it's my pleasure to present this check from Golfers Against Cancer to Dr. Ittman and to the Prostate Cancer Foundation. Here you go.
Thank you, sir.
Projects that you've been helping me fund in retinoblastoma. First of all, retinoblastoma, the initiating event is known. It's mutation in the appropriately named retinoblastoma gene, or RB1 gene. And we now know that that's not sufficient in order to result in cancer in retinoblastoma. There have to be other events that occur for that to happen. We don't know what they are yet. So what we did was that we took retinoblastoma tumors, some of which have the propensity to be able to metastasize and some that don't, and we compared the gene expression profiles between them. And we've come up with about 50 candidate genes that look like they're only expressed in metastatic or invasive retinoblastoma and not in non-invasive retinoblastoma. So we're trying to determine which one of those might be important. And that's probably not just important in retinoblastoma, at least we theorize that that's the case, because other cancers that more commonly happen in adults, the aggressive cancers that have metastasized, frequently have RB1 mutations. And so what is happening subsequent to those RB1 mutations may not just be important in understanding retinoblastoma, but these other cancers as well.
All about Dr. Lee, which is fantastic, because if he was here, he's very humble, and I can be somewhat more effusive about his interesting and amazing research. I also just want to say, on behalf of a lot of the younger researchers that are working with these really distinguished PIs here, that a lot of the money that organizations like the GAC give is helping to develop our careers with some of these smaller projects to eventually allow us to become independent and get funding. So your money and your donations are not only appreciated by the big researchers, but so many of the younger up-and-coming researchers in their labs. So on behalf of myself as one of those up-and-coming researchers that's new to the field, I just want to thank you. And Dr. Lee also obviously wants to thank you as well. It's also very exciting as a young researcher to see how passionate everybody is about their particular tumor of interest. Our particular tumor of interest is childhood brain tumors. With increasingly aggressive therapy, pediatric brain tumors still are the leading cause of pediatric cancer death. And in the children that survive, they often have lifelong complications from their treatment, which usually includes surgery, radiation, and relatively high-dose chemotherapy. So what Dr. Lee's done is develop these mouse models. And he's taken samples obtained from all over the world of 9 different types of pediatric brain tumors, and he implants them directly into the same site in the mouse brain that the tumors occur in the patient brain. So that's what those strange words, the orthotoptic xenograph models mean, is that he takes them and puts them in the same location in the mouse brain. And so he has the largest model system in the world and is using that in various collaborative institutions through the NIH and a lot of smaller brain tumor consortia.
It is my pleasure to present y'all with the check from the Golfers Against Cancer to the Texas Children's Hospital.
Thank you very much.